Bismillahirrahmanirrahim. Assalamu alaikum students. Today we are going to study an important current topic, that is mechanism of antibiotics. Mechanism of antibiotics. Students, the proper knowledge of mechanism means how the antibiotics work in the human body. This study is very important for the selection of antibiotics for the good treatment of a patient. Now, first of all, what antibiotics do when we intake antibiotics? First of all, cell wall synthesis inhibitors. Students, cell wall synthesis inhibitors mean that antibiotics act by stopping the synthesis of cell wall of bacteria. By inhibiting the cell wall of microorganisms, that is bacteria, antibiotics prevent the replication and spread of bacteria. Okay? Cell wall synthesis inhibitors. These antibiotics include penicillins, vancomycins, carbapenems. These class of antibiotics, they prevent the cell wall of bacteria. Now, second mechanism of action of antibiotics is DNA synthesis inhibitors. Means there is a class of antibiotics which prevent. Students, DNA synthesis inhibitor means there is a class of antibiotics which prevent the synthesis of DNA of bacteria. When there is no DNA of bacteria, bacteria will ultimately die off. They will not replicate and ultimately the disease will stop. What are those antibiotics? DNA synthesis inhibitors. For example, fluoroquinolones. Antibiotics belonging to the group of fluoroquinolones, they inhibit the synthesis of bacterial DNA. Now, RNA synthesis inhibitors. Students, these antibiotics, they stop the synthesis of bacterial RNA. When there is no RNA, there is no production of bacterial proteins. As a result, there will be no replication of bacteria. Examples of RNA synthesis inhibitor antibiotics: isoniazid or INH, which is a main anti-tuberculous drug. Now students, protein synthesis inhibitors. These antibiotics, they prevent the synthesis of bacterial proteins by acting on ribosomal subunits. Either they will act on 30S ribosomal subunits or 50S ribosomal subunits. Examples is tetracyclines, chloramphenicol, aminoglycoside antibiotics. Now, DNA gyrase. Students, DNA gyrase is an enzyme which is present inside the bacteria, and this enzyme is important in the coiling and uncoiling process of stranded DNA of bacteria. Antibiotics stop DNA gyrase enzyme. Okay, students? Examples of such antibiotics is quinolones. Now, number 6 mechanism of action of antibiotics is mycolic acid inhibitors. Students, what is mycolic acid? Mycolic acid is present in the cell wall of Mycobacterium tuberculosis bacteria. And anti-TB drug like rifampicin, INH, they act on the mycolic acid in the outermost boundary of Mycobacterium tuberculosis and prevents their replication. Okay? Now, last mechanism of action of antibiotics is folate antagonist antibiotics. Students, there is a class of antibiotics which kills the bacteria by acting as antifolate. Folate is essential in the biological activities of bacteria like DNA and RNA synthesis. Okay? Example of folate antagonist bacteria, they include trimethoprim and sulfamethoxazole combinations, which is commonly called as Septron brand name. Trimethoprim group of antibiotics, they act as folate antagonist. They inhibit the folic acid in bacteria. Okay, students, this was brief about mechanism of action of antibiotics. Students, there lies a question. Why we called antibacterial antibiotics and antiviral antibiotics? This is because mainly these structures are present in bacteria. So, antibacterial antibiotics will act on these structures and inhibit the replication of bacteria. As these structures, cell wall, mycolic acids, folate, these are not present in viruses, so there is no use of antibacterial antibiotics in viral infections. Okay, this was all about mechanism of action of antibiotics. Thank you, students. Allah Hafiz.